The cell membrane is frequently characterized as a static shell, a biological plastic bag keeping the inside in and the outside out. If we push past the extracellular matrix and look at the boundary of a single cell, we find a turbulent, undulating ocean of moving lipids. The fluid mosaic model describes this boundary as a highly regulated semi-solid fluid. It operates as a strict thermodynamic gatekeeper, controlling the exact flow of matter. To understand the mechanics of the membrane, we must strip away the biological complexity and engineer it from scratch. This diagram shows a single phospholipid molecule. It consists of a hydrophilic, water-loving head attached to a hydrophobic hydrocarbon tail. Because the environment is mostly water, these tails hide from moisture. They spontaneously self-assemble into a dual-layered sheet, pointing tails inward. The composition of these tails dictates membrane behavior. Saturated fatty acids have straight chains. They pack tightly, decreasing overall fluidity. Unsaturated fatty acids contain double bonds, creating sharp kinks. These kinks push neighboring lipids apart, restoring rapid, fluid motion. Living cells experience fluctuating temperatures, which threaten to either freeze or melt this delicate lipid matrix. To survive, the membrane relies on cholesterol. This animation illustrates cholesterol molecules inserting themselves between the lipid tails. At low temperatures, they act as physical spacers to prevent rigid crystal formation. At high temperatures, their rigid ring structures restrict excessive chaotic lipid movement. The lipid bilayer is an actively self-assembling matrix. By altering its ratio of saturated fats and cholesterol, the cell tunes its structural fluidity to survive a shifting thermodynamic environment. If the lipids form the structural wall, embedded proteins act as the specialized doors, sensors, and structural anchors. Integral proteins span the entire hydrophobic core, creating a physical bridge between the inside and outside. Peripheral proteins attach only to the surfaces. While lipids and proteins move laterally with ease, flipping vertically requires forcing a polar head through the nonpolar core. This is energetically highly unfavorable. To bypass this energy barrier and maintain a specific distribution of lipids on each side, the membrane utilizes specialized enzymatic proteins called flipases. Precisely arranging specific lipids and proteins on the inner versus the outer layer establishes the asymmetric, highly selective biological machinery of the cell. Every molecule that enters or exits this machine is governed by a universal rule. Movement is strictly dictated by concentration gradients and entropy. In passive transport, particles move spontaneously from high concentration to low concentration. The Gibbs free energy change is negative, meaning potential energy dissipates naturally without the cell performing work. Small nonpolar molecules like oxygen slip directly through the lipids in simple diffusion. Large or charged molecules are blocked by the core and route through protein channels, known as facilitated diffusion. When a solute is entirely blocked from crossing the membrane, the thermodynamic pressure to equalize the system remains. The solution to this imbalance is osmosis. This equation represents osmotic pressure. Osmotic pressure is a colligative property. It depends only on the number of dissolved particles, not their identity. You can think of it as a sucking pressure. Because the solute cannot move, massive volumes of water are pulled across the barrier to dilute the trapped particles. Passive transport and osmosis are the inevitable thermodynamic drive toward chemical equimolarity. Left unchecked, they will eliminate all gradients, establishing an equilibrium that the living cell must constantly fight against. How does a cell import necessary nutrients when the concentration gradient is entirely against it? The answer is active transport. The cell utilizes stored energy to force molecules uphill, generating a positive change in free energy and pushing the system away from chemical equilibrium. In primary active transport, the cell hydrolyzes an ATP molecule. The energy released powers a transmembrane pump, driving the solute against its gradient. Secondary active transport uses a different mechanism. The downward flow of one particle is harnessed to drag a completely different particle up its gradient, either in the same direction, symport, or opposite directions, antiport. Active transport is the cellular equivalent of rolling a boulder up a steep hill. The cell intentionally fights equilibrium to store immense potential energy for future biological work. By selectively pumping charged ions across an impermeable barrier, The cell functions precisely like an electrical capacitor. The core engine of cellular electricity is the sodium-potassium ATPase pump. It continuously exports 3 sodium ions out of the cell while importing 2 potassium ions. Because the pump removes 3 positive charges for every 2 it imports, the stoichiometry inherently leaves the cell's interior with a net negative charge. Additionally, the membrane contains leak channels that are highly permeable to potassium at rest. As positively charged potassium constantly bleeds out down its concentration gradient, the negative charge of the cell's interior deepens. To calculate the exact voltage generated by these ion imbalances, we rely on physical chemistry established over a century ago. Equation 8.2 is the Nernst equation. It calculates the theoretical equilibrium voltage for a single isolated ion, based purely on the ratio of its concentration outside the cell to its concentration inside. However, a biological cell is not a single ion system. To solve the whole cell voltage problem, electrophysiologists expanded the model to account for multiple competing gradients. Through collaborative research, biophysicists finalized a master formula that accurately captures the entire electrical environment. Equation 8.3 is the Goldman-Hodgkin-Katz voltage equation. It scales Nernst values by incorporating the relative permeability, represented by P, of sodium, potassium, and chloride simultaneously. Notice a mathematical quirk. Because chloride carries a negative charge, its internal and external concentrations are inverted in the numerator and denominator relative to positive ions. The standard resting potential of a neuron, typically around -70 millivolts, is the strict mathematical output of thermodynamic gradients actively maintained by the lipid mosaic. While this generalized model dictates the plasma membrane, specialized organelles actively alter these rules to perform specific biological functions. If we plunge rapidly from the outer boundary, traveling deep into the busy cytoplasm of the cell, we reach the mitochondrion. The outer mitochondrial membrane is highly porous, but the folded inner membrane, the cristae, is severely restricted. This diagram shows a cross-section of that inner membrane. To generate the massive proton motive force required to synthesize ATP, the inner membrane alters its lipid composition. It contains high levels of cardiolipin and entirely lacks cholesterol, resulting in extreme impermeability to protons. We have built a dynamic, self-assembling fluid boundary from first principles. It weaponizes thermodynamic gradients to calculate precise, life-giving voltages. The fluid mosaic model is the tunable bedrock of physiology. Mastering the physical chemistry of this thin film of fat explains the mechanics of human life, from deep neural thought to cellular